day, everyone. Today, we're going to tackle about ligase chain reaction. So, let's head on its introduction.
The invention of the polymerase chain reaction in 1985 paved the way for the development of virus nucleic acid detection systems. These aided scientists to identify genetic and infectious diseases, discover novel genes, and unravel the mysteries of protein ligand recognition. Among these DNA amplification methods, ligase chain reaction was developed as a complement or alternative for PCR. So what is ligase chain reaction? This is a type of probe amplification technique that involves two enzymes, DNA polymerase and a thermostable ligase, which helps join four oligonucleotide probes together, followed by standard amplification using polymerase chain reaction or thermal cycling. This technique was proven to be cost-effective, simple, and robust with a high degree of sensitivity and specificity in addition to multiplexing ability.
You may be thinking, ligase chain reaction sounds familiar to the polymerase chain reaction, another DNA amplification technique. But the main difference is that the polymerase chain reaction amplifies a segment of DNA, whereas the ligase chain reaction amplifies a short DNA probe. Unlike PCR, it only uses Taq polymerase. LCR uses both DNA polymerase and DNA ligase. PCR amplification exploits two primers to obtain three types of information. First is presence of target sequence, second, distance between primers, and third, sequence in between the primers. While LCR exploits four primers to obtain only two types of information, the presence of adjacent target sequences and presence of perfect complementarity to the primers at the junction of these sequences.
The principle of LCR is based on the ability of DNA ligase to join two complementary probes during hybridization of the target molecules. Newly ligated oligonucleotides will serve as the DNA template for further annealing, with each cycle yielding double result of the target of interest.
B. Ligase chain reaction steps.
In an LCR mixture containing four oligonucleotide probes, DNA ligase, DNA polymerase, and NAD+ are added to the clinical sample. LCR reactions are initiated by heating the mixture at 94 degrees Celsius to separate the double-stranded DNA. The mixture is then cooled down to about 40 to 60 degrees Celsius in order to facilitate the hybridization or binding of the four oligonucleotide probes to the target DNA strands. Subsequently, the gaps of the adjacent probes are filled by DNA polymerase, and the nicks are covalently joined by thermostable Taq ligase. Once the probes are sealed, these short DNA segments will then be used as a DNA template, and succeeding cycles will produce logarithmic amplification of the target DNA. The radio-labeled LCR products can be visualized electrophoretically on denaturing polyacrylamide gels and autoradiography.
Now we're going to talk about how significant the application of ligase chain reaction is in diagnostic medicine.
The LCR has been used for genotyping studies to identify the presence of specific genetic disorders such as sickle cell disease, wherein discrimination between normal beta-A and sickle beta-S globin genotypes in humans are achieved using either isotope detection method or fluorescein-labeled LCR primers. It has now become widely used in infectious disease detections, both in diagnostic and research settings for bacterial and viral pathogen-borne diseases. It can also detect other target sequences such as single-base permutation in HARAS proto-oncogene, G6PD, and HOXB7. Provided suitable primers are available and there are easily obtained specimens such as urine that can be used for diagnostic purposes, screening of large numbers of people is practical. This can very well facilitate research for better understanding of the epidemiology of specific diseases.
Lollipop, lollipop, oh, lolly, lolly.